Okay, this video goes along with the notes, just the last set of notes on Punnett squares, and this is sex-linked and dihybrid crosses. Okay, a couple of things to understand about chromosomes before we get started. Typically, humans have one pair of sex chromosomes, also known as gametes. So gametes are sex chromosomes. Our egg and our sperm are gametes, which are sex chromosomes. Also, we have 22 pairs of what are called autosomes, or somatic chromosomes. These are the chromosomes that code for our height and our hair color and our skin color and all of those traits except for our gender for the most part. So total we have 23 pairs, or we have 46 chromosomes. So when you're looking at problems and they're asking for the number of pairs or the number of chromosomes, again, make sure that you are reading the problem carefully. Okay. Female chromosomes, okay, are represented by the alleles XX. Male chromosomes are represented by the alleles XY. The X chromosome is significantly bigger than the Y chromosome. Therefore, because it's bigger, it can put more stuff on it. It's going to carry more information than the Y will. The Y basically determines the gender. So you could be an XXXXXXY, and you're still going to be a guy. You could just be a single X, but you're still going to be a girl because you don't have a Y chromosome. And we use karyotypes to compare and look at our chromosomes. They literally take a picture of the chromosomes, and they cut them all apart, and then they pair them up. And you can see, you can match these up. They're by size, by the banding pattern here. This is showing the different genes. And you can see we have 22 pair. And then we say this last spot here, the 23rd pair for our gametes. So you're either going to have an X and a Y if you're a normal guy, or you're going to have two X's if you're a normal girl. Since females have two large X chromosomes, they have duplicate information. If you remember back when we did cell cycle, we talked about homologous chromosomes, how they're coding for the same traits, but they're not identical. Well, the same thing applies here. They're coding for the same traits, but they're not identical. So since there's a lot of genetic disorders that are carried on the X, and they're recessive, females are less likely to show the disorder. So again, remember, recessive alleles is just like the basic Punnett squares. If you are recessive, if you have two recessives, you're going to show the trait. If you have a dominant and a recessive, you're not going to show the trait. Therefore, females have a backup plan. Okay, so if we have one that's not correct, we have something else that will cover up for it. Since males only have one X chromosome, all that information is going to be expressed. They don't have a backup plan. Even if there's a mutation on that X chromosome, it is still going to be seen. Therefore, males have a much greater possibility of carrying or expressing a genetic disorder than females would be. Okay, so how does this apply to Punnett squares? So when we're doing our Punnett square, the genotype or the genes of my parents have to show both the gender and the trait. So they're going to have two letters instead of just one. So if we're going to look at color blindness, because that's a real common one, we know it's sex-linked, we know it's recessive, and we're going to call it the letter A. So if you do not, if you are normal, you're going to be a big A. If you do have it, it's going to be a little a. So if we're talking a female who's colorblind, she would have X little a, X little a. She has two X's because it's a female. She has two recessive alleles because we know that she is colorblind. And again, A is the colorblindness trait in this particular example. If you're a normal female, you're going to be X big A, X big A. The thing is, females can be heterozygous because we have two alleles here. So if you are heterozygous, you're going to be called a carrier. And your alleles would be X big A, X little a. And the thing is with this, you can pass it on to your offspring, but you don't have that particular disorder yourself. If you're a male and you're colorblind, your letters are going to be X little a, Y. Again, the X and the Y represent the male. The little a says you have the disorder because it's a recessive disorder. If you're normal, you're going to be X big A, Y. And males can't be carriers because they only have one X. So what you see there is what you're going to see as being expressed. Okay, so let's do a couple of practice problems. These again coincide with the worksheet. And it says, number 1, that normal vision is dominant, or big B, while colorblindness is recessive. And that's a typo on my part. After recessive, that should be a little b, not a big B. So it says, cross a female with normal color and is not a carrier with a man who has color blindness. Well, the first thing we're going to do, whenever you're talking males and females, is we're going to put up the X's and the Y's. So here's mom, and here's dad. Okay, it says mom is normal. Okay, so if she's normal, that means she's got to be big B, little b. Excuse me, big B, big B. Because we just said that normal had 2 dominant alleles. Our man is colorblind, so he's going to be little b. Because that's a recessive allele, shows that he has that trait. Okay, we're going to do our Punnett square just like we've always done them, with one exception. This here is an allele. This is another allele. So this allele is going to go here, and this allele is going to go here. Same with dad. I have my recessive allele going with my X, and then we just have the Y. And remember, the Y doesn't have this because it's so tiny. Okay, we're going to carry everything down. We're going to carry everything across, again, just like we've been doing. In this case, we're going to carry the Y's across. And that's our probabilities for our offspring. So let's figure out the genotype and the phenotype of the offspring. There's genotype, there's phenotype. Again, genotype's the letters. So we look to see what's in the boxes. I've got an X big B, X little b. I've got one there, and I've got another one there. So I've got 2 of 4. I've got an X big B, Y, and an X big B, Y. Again, I've got 2 out of 4. I know I've got 2 X's here, so I know in my phenotype or my description, this is going to be a female. And she has a big B and a little b, which makes her a carrier. Again, she's not colorblind, but she can pass it to her offspring. Down here I have X and Y, so I know it's a male. He has a big B, that is the dominant allele. He's going to be normal. Okay, let's do a couple more problems. Again, just to make sure we understand these. Okay, still working with sex-linked Punnett squares. This is number 2. It says, cross a woman who is not colorblind, but whose father was, with a man who has normal vision. So again, anytime you see male and female, you automatically put in the X's and the Y's. Okay, it says that she is not colorblind, so we know we have to have one big B. It also says that her father was. In order for her father, if her father's colorblind, he's going to pass his allele onto the daughter. Since he only has one X, she got one of those X's from dad. So we know that she's got to be a carrier. And we're going to cross her with a man who has normal vision. Again, normal vision, big B. Do the Punnett square. Separate my alleles. Carry everything down. Carry everything across. And then figure out the genotypic and phenotypic probabilities of our offspring. Here's my genotype. Here's my phenotype. Look at my boxes, see what my letters are. I've got an X big B, X big B. any more. I've got an X big B, X little b. Again, X big B, X little b is the exact same thing as X little b, X big B. I just like to put the dominant alleles first. I've got an X big B Y, and I've got an X little b Y. Okay? So I've got a 1 out of 4 in all of these. Okay, let's figure out what they look like. What's our phenotype? Okay, I got 2 X's, so I know it's a female. I have 2 big B's, that means she's going to be normal. I have 2 X's, so it's another female. She has a big B and a little b, so she's a carrier. I have an X and a Y, so I know it's a male. He has a big B, so he's going to be normal. I have another X and a Y, so I know that this one's also male. He's got a little bit, a lowercase b or little b. He's going to be colorblind. Okay, last problem for sex linked. It says hemophilia or bleeder's disease is a recessive sex-linked disorder. Now, hemophilia is like if you get a cut, your blood won't clot, and so you just keep bleeding. And sometimes it could be internally. Sometimes if you fall and cause some breaks on the inside of your body, you can bleed to death just inside. So, but again, it's another recessive genetic disorder. I'm going to do it the exact same way. Okay, it says cross a female who is a carrier for hemophilia with a man who has normal clotting. Okay, again, I see man, I see woman. First thing I'm going to do is I'm going to write X, X, cross X, Y. Okay, it says my woman is a carrier. So she can be big H, little h, because we know carriers are heterozygous. We say that my man is normal. So he's a big H. Okay, do our Punnett square. Separate my alleles, so I got an X big H and an X little h. And then I've got an X big H and a Y. Carry everything down. Carry everything across. That's my Punnett square. Again, let's figure out the probabilities of my offspring. Here's my genotype. Here's my phenotype. Look at my letters in my boxes. I got an X big H, X big H. I like to make little tick marks because then I know I've counted them. I've got an X big H, X little h. I've got an X big H Y. And I've got an X little h y. And all of these are 1 out of 4. My 2 X's tell me it's a female. She has 2 big H's, so she's normal. I have 2 X's, so it's another female. I have a big H and a little h, so she's a carrier. Again, she doesn't have it, but she can pass it on to her offspring, so she can carry it on to the next generation. I have an X and a Y, so this is a male. He has a big H, so he's normal. I have an X and a Y, so it's another male. He has a little h, so he has hemophilia. Okay, so that's sex-linked. Now let's go for the really fun ones. And this is dihybrid. Now, anytime you see the prefix di, you know that we're dealing with 2. Okay, so that kind of gives you a clue. We're going to be looking at 2 traits instead of just 1. And we're going to assume for this class that everything is going to be dominant and recessive alleles. We're not going to have different letters. We're just going to have uppercase and lowercase. Okay. Again, we talked about when we started this about the law of segregation and the law of independent assortment. And we know that the segregation or separation of one gene will not affect the separation or segregation of another. Okay. And this leads to more genetic diversity, because each chromosome has lots and lots of genes. So, yeah. So to determine the segregation of the parents' alleles, we're going to use the FOIL process that you've learned in algebra. Okay, and just a reminder what FOIL is the first, outer, in, and last. So first is the first letter of each gene. Outer is the outer letters of the gene. Inner is the inner letters. And last is the last alleles of each gene. And you can use that and you can come up with something like this. So let's do a practice problem. Okay, it says cross a pure green-leafed, wrinkled seed plant with a yellow-leafed hybrid round seed plant. Green-leafed and round seeds are dominant. So we've got 4 traits that we're looking at here. We're looking at green and yellow. So we're looking at color. And we're looking at wrinkled and round. So what does our seed look like? So we've got wrinkled and we've got round. Okay, now it tells us, right in the problem, that green and brown are both dominant. So green is going to be my uppercase letter. In this case, I'm going to use the letter G. Yellow is not going to be Y because it's recessive. It's going to be little g. Wrinkled and round, it says round is dominant. So my round is going to be big R, and my wrinkled can be little r. Okay, so that's the traits we're looking at. The genotype of the parents. Okay, so we have to figure that out. So it says it says pure green leafed. So pure green leafed, it's going to be big G, with a wrinkled seed. So that's going to be little g, little g. And we're going to cross that with a yellow leaf. We said yellow was little g. Again, I can't have a big G here because otherwise it wouldn't be yellow. And hybrid round. Now hybrid means different. So we've got a big R and a little r. So if we use our FOIL, here are the combinations for my parents. This is parent 1. Sorry, these are little r's. This big G can go with this little r. Or this big G can go with that little r. This big G can go with that little r, or it can go with that little r. Same thing here with my second parent. This big G can go with the little g can go with the big R. This little g can go with the little r. This big G can go with the big R, or that little g can also go with the little r. So those are the possible allele combinations for our parents. Now, taking all this information, we're going to make our Punnett square. These Punnett squares are big. They're 16 squares. So draw a big Punnett square and split it in half a couple of times. And what you end up with is 16 squares. Okay, so we said parent 1. We go back and we look. Parent 1, all of them were big G, little r. So we're going to put a big G, little r at the top of each column. We said our other parent could be little g, big R, or little g, little r. So little g, big R, Add a little G little r. Add another little G big R and another little G little r. Again, we're going to carry everything down. So we're going to end up with 4 letters in each box. So we have a G, R. I'm just going to carry them down. All of these. And I didn't make my squares quite big enough, so that's why I'm going on the line. It's a little better. Okay. Yeah. That's just an ugly R. Okay, I'm going to carry everything across. So we've got G, big R, all the way across in this row. I've got G little r that I'm going to carry all across in this row. I've got another G big R. And I've got another little G little r. And that is the probability of my offspring. So let's put this together and make some sense out of it. Again, here's my genotype. Here's my phenotype. When I look at this, I see I have a big G and a little g and a big R and a little r. Now, you want to put the G's and the R's together at this point because it's going to make it easier to determine your phenotype. So I've got a big G, big R, little r. And if I start looking at the rest of my squares, I can see I've got one here, and I have, that's an R, and I have 2 more down here. So I have 4 out of 16 that are big G, big R, little r. I come over here, I see I have a big G, big R, little r. Something is messed up. Oh, no, I know what the problem is. Okay, this is a different problem than I did other. Okay, so I don't have 4 out of 16. If I look, I've got big G, I've got more of them over here. And that's why I like to put little check marks in the boxes so I know what I've done. So I've got a big G, big R, little r. I've got 2 more here. So I have 8 out of 16 that are big, little. If I look here, I've got a big, little. So I've got a big G, little r, little r. And I've got 1 there, I've got 1 there, I've got 1 there, 1 there, and 4 more down here. So I've got 8 out of 16 here. So then I go back to my phenotype. What's my description? Big G, little g. It's got a dominant allele, so I know that this one's going to be green. And I got a big R here, so I know it's going to be round. On this one here, I still have a big G, so I know it's going to be green. But I have 2 little R's, so I know that's going to be wrinkled. Okay, on the last page, we're just going to set this problem up. We're not going to do it all the way. It says, cross a hybrid spotted purple alien with a striped heterozygous orange alien. So my traits are spotted and striped. These are actually the same trait, there's just different alleles for that. And we have purple, and we have orange. It doesn't doesn't tell us what's dominant and what's recessive, but we can figure that out because it says hybrid spotted. And in order for hybrid to be seen as spot, we know that spotted has to be a dominant allele. And I don't like the letter S's because I can't tell if it's a big S or a little s, so I'm going to use an A. So spotted is a big A, striped is going to be a little a. And then it tells us it's a heterozygous orange. Again, heterozygous is different, so that means orange here has to be my dominant allele. That means purple is recessive. Okay, so the genotype of the parents is a hybrid. Hybrid's different. Hybrid spotted, so big A, little a. Purple, we just said purple was recessive, so there's my first parent. Parent number 2, striped. In order to see striped, I can't have a dominant allele, so it's got to be 2 little a's. And heterozygous. Heterozygous means different, so we got a big B, little b. Okay, again, using FOIL, we're going to come up with the combinations. My big A can go with my little b, or my big A can go with my little b, or my little a can go with my little b, or it can go with that little b. The genotype for parent 2, again, using FOIL, I know that little a can go with the big B, that little a can also go with the little b, this little a can go with the big B, or this little a can go with the little b. And then we're going to set up the Punnett square just like we did on the last one, and then figure the genotype and the phenotype of our offspring. And that is the last set of notes on Punnett squares. It's the funnest set of notes because it's the most challenging.